So now that we have looked in depth at the process of mitosis, we want to look at a different process that is related but has a different purpose, and that is that of meiosis. Meiosis is a special type of cell division that actually produces our sex cells. We refer to these as gametes, and that means sperm and egg. It is also known as what we call reduction division because the chromosome number gets cut in half. This is involved in sexual reproduction. So let's go through some general vocabulary here, and then we're going to do the chart that actually kind of takes us through the process of meiosis before you do your lab. So the first piece of vocabulary is what we call homologous chromosomes. Now, you've seen this prefix before, homo, which means same. So these chromosomes are the same. These are two chromosomes found in each body cell, somatic cell. And how they are the same, they are same in size, they're the same in shape, and they're also same for the genes that they code from. You get one of these from mom and one of these from dad. The second piece of vocabulary is diploid. Some people call it diploid, I call it diploid. That describes a cell that contains pairs, so it has both mom and dad's homologous chromosomes in there. We refer to this as the 2N, so where it's the number of chromosomes. This again is in somatic cells. Humans have 23 pairs of chromosomes, so our diploid number is 46. By contrast, haploid kind of sounds like the word half, and that's exactly what it means. It's a cell that has half of the diploid number of chromosomes. So therefore, we only have one set of genes, and so instead of being 2N, we refer to it as N. These occur in the gametes, or the sex cells, and for humans, our haploid number is 23. The last piece of vocabulary that we'll look at in this section is crossing over. And this is basically where a homologous pair of chromosomes exchange segments. Now this can happen in mitosis, but it's exceedingly rare. It occurs during a process called synapsis that we'll get to here in a second during prophase 1 of meiosis. And this is important because it increases variation in our offspring because we get new combinations of genes. So let me show you this process here. So I've got chromosome 1 and chromosome 2, and they literally come together and they switch. So now the 3 genes that were on the gray one are now on the white one, and the 3 genes that were on the white one are now on the gray one. All right, so we are now going to get into the chart that kind of walks us through the different stages of meiosis, or the phases of meiosis. So the first stage is prophase 1, so that goes in the top box there. And we have a couple of definitions. First one is synapsis. And synapsis occurs when the homologous chromosomes actually paired up. Okay, so you get pairing up of moms with pairing up of dads. And we also have something called a tetrad. And a tetrad has 4 chromatids. So in terms of the events in the stage, so this is a tetrad. So we've got 4 chromatids, and so we have a tetrad. In terms of the events, okay, the first event is, of course, synapsis, and we also get crossing over. In this stage, the cells are diploid, and the chromosomes are double-stranded. So what does it mean that they are double-stranded? Well, when we have a chromosome that looks like this is what is referred to as a double-stranded chromosome. In other words, it has 2 chromatids. So let's go ahead, and in the picture of the stage, we're actually going to draw this. So I'm going to actually draw a cell here, okay? And in it, we're going to put 2 chromosomes. We're going to put We'll make the red ones here. Let's say these are from mom. We have a big one. We have a little one. And then the blue ones, actually that doesn't show up very well. Let's do yellow. There we go. Are from dad. So we have a big one. And then we have this little one. Okay, so the big ones, they're homologous, they pair up, and the little ones. So remember, again, we have a tetrad here. So you want to color this using colored pencil. You can actually make them 4 different colors if you wish as well. All right, so the second phase is metaphase 1. And in metaphase 1, we have independent assortment. So the chromosomes are going to line up along the equator kind of randomly. All right, so the homologous chromosomes they line up across the equator from each other, and you will see that in the picture. The cells here are diploid, and the chromosomes are double-stranded. So let's go ahead and draw this. Okay, so again, we have our cell, and this time I'm actually going to put a dotted line down the middle. This represents our equator. And what we're going to do is we're going to have our chromosomes line up across from each other. Now, this is random. Okay? It's not that all of mom's chromosomes line up on the left-hand side and all of dad's chromosomes line up on the right-hand side. That's not how it works. Okay? Actually, I think next time we're going to just make these X's. Starting on the next slide, because these curlicues are tough. All right, so they line up across, but notice that their centromeres are not on the equator. So this looks different than mitosis. We then move to anaphase 1. And in anaphase 1, we get disjunction, which is where the homologous chromosomes actually separate. So in terms of events, we get disjunction. And again, the cells are diploid, and the chromosomes are double-stranded. So what happens here in our picture, we have our cell. Okay. We have our equator. And I have this chromosome here, this chromosome here, and then this chromosome here, and this chromosome here. And what happens is they move and they separate. Okay, this is what we call disjunction. In telophase, so here's an animation actually of disjunction. So you get them lined up across the equator from each other, and they actually pull apart and they separate. So that is the process of disjunction. The next phase, and the last one on this side, is telophase 1. In telophase 1, we get cytokinesis into 2 cells. The cells now change from being diploid to being haploid, but the chromosomes are double-stranded. So, what does this look like? Well, what happens is, basically the cell membrane pinches in at that equator. So I end up with 2 cells. I have a big mom and a little mom. Oops. Oh, there we go. And then I have the big one from dad and the little one from dad. Okay, so my diploid number was 4. Now each cell only has 2, but the chromosomes are double-stranded. Now, we're going to go through a second division here, but in this case, we won't replicate the DNA before it happens. So the second phase is prophase 2, and here there's not a lot going on. The chromosomes just start moving towards the center. The cells are still haploid, and the chromosomes are still double-stranded. So in terms of your picture, it's not going to look a whole lot different than telophase 1. We're still going to have our 2 cells. We're going to have the big one and the little one, and they're just slightly moving towards the center. It's kind of hard to draw this particular event because there's not a lot going on necessarily. All right, in metaphase 2, the chromosomes line up along the equator, and this time they're going to line up so that their centromeres are actually on the equator. The cells are haploid, and the chromosomes, I should say not doubles, they are double-stranded. All right, so this is what, go back here. This is what you would see in each cell. So you'd have your one cell here, and your second cell here, with the equators. Okay, and this one big and little. Okay, so the idea here is the centromeres. So this looks very much like metaphase in mitosis. It's just the cells are haploid at this point. All right, anaphase 2, we get the chromatids get pulled apart and they separate to opposite poles. So again, very much like mitosis. The cells are now diploid because we have 2 chromatids, so that doubles the number. And the chromosomes are single-stranded. So this is what it's going to look like. We have our cells with our equators. Okay, so we have half and half, and a little half and a little half, and then we have half and half, big half, big half. And they are getting pulled to either end. The last one, oh, so here we go. So this is kind of what is happening. Okay, they actually get pulled apart just like this. All right, so we have a single strand, so it is now 1 chromatid, but because we have 2 of them, they each count as a separate chromosome, so that's why we temporarily here go back to diploid. In telophase 2, we get cytokinesis of both cells, so now we get 4 cells. So the cells are, what happened there? Cells are haploid, and the chromosomes are single-stranded. So this is what we would see happening. We've got our 4 cells. Each of them has 2 chromosomes. Okay, so they are haploid, and those actually go ahead and go through a maturation process to become sperm or egg.